Welcome all of you. It's Friday, 8:30. We're celebrating vision research. It is the most exciting time of the week. Maybe somebody will pay attention. No? Are we going to pay attention, Cher? All right. So we have everybody now focused. And the focus is needed because we have today a speaker from a different state. That's like a different country. It's from far away. And we're truly delighted to have him today. And again, everybody here and on a Zoom, welcome. And let's introduce our speaker.
Good morning, everyone. My name is Sam Du, and I'm a PhD student in the Paczewski Lab. And it's my great pleasure today to introduce a physician scientist who's doing fascinating work on photoreceptor diversity and therapies, Dr. Joseph Corbo, MD, PhD, to our distinguished speaker series for his talk titled Spectral and Cellular Reprogramming of Photoreceptors. Dr. Corbo is currently a professor of pathology and immunology at the Washington University School of Medicine in St. Louis. He received 2 bachelor's degrees from Stanford University in linguistics and biological sciences before completing his MD PhD at UCSD, studying development with his thesis advisor, Mike Levine. He then pursued his residency in anatomic and neuropathology and postdoctoral research at Brigham and Women's Hospital and Children's Hospital at Harvard Medical School, where he was mentored by Chris Walsh and Connie Sepko. After completing his training, he joined the faculty at WashU, where he has run his research program and clinical practice ever since. He's a clinician scientist who utilizes neurobiology, cell biology, and genomics approaches to investigate the development and function of the retina and the CNS. In particular, he's interested in biochemical and transcriptional mechanisms that contribute to photoreceptor diversity. He does not only study mouse and human. Some of his most fascinating work has described color patterning and photoreceptor diversity in birds. He's heavily involved in teaching at the undergraduate and graduate levels at WashU, and is, of course, very well published with over 87 peer-reviewed papers in journals such as Science, Nature, and Cell, and with the H index of 47. Please join me in giving a warm welcome to Dr. Corbo.
Well, I've never had an introduction like that. It's a real pleasure to be here. I want to just highlight 2 connections I have to UCI, one longstanding and one of the more recent vintage. So I grew up in Newport Beach, spent my summers surfing down on the peninsula, and went to high school at Newport Harbor High School, where my best friend, his older brother, was attending UCI at that time in the mid-1980s, and he had a radio show on KUCI. Actually, believe it or not, he still has that radio show on KUCI. You can listen to it. But we used to go on KUCI in the 80s and play live music, very bad live music. So those recordings still exist, but I won't let you hear them. And then the second and more recent connection is with Vladimir Keflov, who was a longstanding friend and collaborator at WashU, and our collaboration continues. And that's why I chose the 2 topics to talk about today, because they are reflective of our longstanding collaboration. And so in the first part, I'm going to tell you briefly about a somewhat older story, just to give you an idea of the sort of non-mammalian, more basic aspects of the research in my lab about spectral reprogramming in a natural setting. And then in the second part, I'll tell you about some more recent and unpublished work where we try to use cellular reprogramming of rod photoreceptors to confer a neuroprotective effect in models of photoreceptor degeneration. And so, as everyone always says at these meetings, you don't need this introduction, but we all know that when light enters the eye, it must pass through the layers of the retina to reach the photosensitive outer segments of the photoreceptors, where the light signal is converted into an electrical signal processed by the retina and then returned to the brain as visual information. There are 2 main types of photoreceptors in the retina, the rods and cones. The rods primarily responsible for dim light vision, and cones for bright light vision, color vision, and high acuity vision. And the real business end of the photoreceptor is the outer segment, which houses the visual pigment, which again, as we all know, consists of the opsin protein and the covalently bound chromophore. And the chromophore that most of us are familiar with, 11-cis retinal, is transformed by absorption of light into the all-trans form of the chromophore, which in turn changes the three-dimensional structure of the protein, initiating phototransduction cascade. But less well known, especially to people who primarily study in mammalian systems, where 11-cis retinal is the only chromophore that's normally used. A lot of aquatic species, particularly those that inhabit freshwater and murky habitats, will often opt for an alternative chromophore, which is derived from vitamin A2 called 11-cis 3,4-didehydroretinal. And the only difference between the two chromophores is the addition of another double bond in the ring here, but that has actually rather profound functional effects on the absorption properties of the chromophore and the visual pigment as a whole, leading to a red shift in the absorption of the molecule. And that's shown here, basically. If you have an opsin with A1, it'll have an absorption like this. If you switch to A2, you get a rather profound red shift. You also get some decrease in photosensitivity. And so the idea is that this red shift, when you switch the opsin, when you switch the chromophore, enables the animal to better perceive longer wavelength light in environments where the ambient light is red shifted. And I think no organism better demonstrates this dichotomy in the visual system than adult bullfrogs, which often hang out at the water surface and use their upper retina to look for interesting bits to eat underwater through a sort of murky aquatic environment, or use their lower retina to view the upper visual field in the aerial environment. And strikingly, if you extract the retinal pigment epithelium from the dorsal versus the ventral, you'll see that the dorsal RP is chock full of vitamin A2, which is entirely absent from the ventral RP. So this animal has really shown that within a single retina, you can sort of specialize the different parts of the retina for different types of vision. This kind of change can also occur dynamically in the life of an organism. So we like to use the example of salmon, which when they're living in the open ocean, primarily use an A1-based visual pigment. And then when they decide to move up into murky inland streams to spawn, they'll dynamically shift to an A2-based pigment. And so a number of years ago, we got interested in this question that had long been posed, and that is, what is the actual mechanism or the identity of the enzyme that converts the 11-cis retinal or vitamin A1 pigment into a vitamin A2 pigment by the addition of this double bond? And I won't go into all the details, but to tell you that we used a couple different model systems, both bullfrog as well as zebrafish, which will undergo this transition when you treat them with thyroid hormone, to do expression profiling and identify an enzyme, CYP27C1, which mediates this transformation. We then made knockouts in zebrafish. We also expressed this enzyme heterologously in mammalian cells and showed that it's both necessary and sufficient to transform A1 into A2. And so I just wanted to finish here in recapping this story by showing you a couple of the functional consequences of removing CYP27C1. And this, again, was work that we did in collaboration with Vladimir and Shinya Sato in his lab. And we had both wild type and CYP27C1 mutant zebrafish. And under normal conditions, they both only have vitamin A1 and don't express CYP27C1. And so when you do single cell suction electrode recordings of the red cone, you see similar absorption in both the wild type and the mutant. But after you treat the zebrafish with thyroid hormone, the wild type will turn on CYP27C1, and that induces expression of A2 and replacement of the chromophore in all of the opsins into visual pigments. And in the wild type case, you get a very dramatic red shift in the absorption of the red cone, almost 40 nanometer red shift, whereas the mutant shows no shift at all, indicating that the enzyme is crucial for turning on and expressing vitamin A2. And so we were really interested in asking the question of what the effect this has on the behavior of the animal, in particular, the animal's ability to respond to light. Adult zebrafish have a positive phototactic response. And so what we did was we would have a light in this sort of arena where the fish is going to be monitored at a very long 940 nanometer wavelength light underneath, a night vision camera above, so we can monitor the location of the fish either in total darkness or in when we are showing it a light from the side here. And so the idea is we shine a light, monochromatic light at different wavelength on the side, and basically see if the fish can respond to it. So if the fish can see the light, it'll swim toward the light and spend more time at this end of the tank, and you can quantify how much time it spends at that end of the tank. And so just to show you what this looks like under conditions when the fish is, when the lights are off, I don't actually know. I'm going to be able to get this to play here. There we go. So the fish will just sort of swim around the tank in dark like this and doesn't necessarily spend more time in one place or another. But if you turn the lights on at one end, then if the fish can see the light, they'll spend a lot more time down at that end of the tank. And this is speeded up a bit. And so that's the sort of setup. And what we used was two different wavelengths of light, either 590 nanometer light, where both the wild type and mutant fish ought to be able to see the light, or near infrared light of 770 nanometers, which we expect. This is the red cone sensitivity of the wild type red shifted fish. This is the sensitivity of the mutant fish. So we expect that the wild type ought to be able to still see this light, but the mutant might not be able to. And so first with the yellow light, whether the animal is wild type or mutant, and whether they've been treated with thyroid hormone or not, they can all increase the amount of time they spend down at the end of the tank where the light is on. They spend about 25% of their time down there in the dark, and that jumps up to between 50 and 70% of the time when the light is on. But in contrast, when you use this really long wavelength near infrared light, only the wild type fish that have been treated with thyroid hormone can see the light and go down there and spend more time at that end of the tank. So this is, I think, a rather striking demonstration of the utility of this system for enabling the animal to dynamically switch or spectrally reprogram their photoreceptors in response to the need to see better in dim, either dimly lit or red shifted aquatic environments. And so this is just a quick summary of what we found. Basically, by profiling, we found this enzyme. It's a member of the cytochrome P450 family of enzymes. It converts 11-cis-retinal into this form with an additional double bond, but it has a fairly profound effect on the ability of an animal to dynamically, spectrally tune their visual system to the environment that they live in. And again, this was a collaboration with Vladimir and Chinya, and two people in my lab who did this work, Jenny Enright and Matt Toomey. Now that was a study that was done a fair bit of time ago, but I want to tell you now a bit of a longer story about cellular reprogramming and our efforts to prevent photoreceptor degeneration in a variety of mouse models. And so probably many of you are familiar with photoreceptor degeneration and how it progresses. This is just an example of a mouse model of retinitis pigmentosa. And what you see is a progressive loss of the cells in the outer nuclear layer over time. And the way it typically works in a rod-dominant animal is you get first progressive loss of the rod photoreceptors with relative preservation of the cones, but invariably there's a late phase of cone loss that occurs even though the gene that's mutated and causing the degeneration is not even expressed in cones. So this is a secondary cell non-autonomous effect on cones. And so this is sort of the before and after of degeneration in a mouse model, where the outer nuclear layer is virtually deleted, but the inner layers, at least initially, are still relatively intact. And if you think about therapeutic strategies for treating photoreceptor degeneration, you can sort of group them into those applicable after the degeneration process is advanced versus those that are applicable before degeneration has progressed. And so afterwards, of course, you could imagine cell replacement therapy, where you say, try to take stem cells, generate a photoreceptor in a dish, and then try to transplant it back into a patient. Alternatively, people are pursuing optogenetic strategies where you introduce a photosensitive molecule into one of the remaining cell populations in the inner retina to try to endow them with photosensitivity. Alternatively, you can take an approach before degeneration has progressed, either standard gene replacement or augmentation therapy. If you have a mutation in gene X, then you try to provide a normal intact copy of gene X, or some sort of more general neuroprotective strategy, where instead of trying to target or prevent degeneration in a specific genetic model, a more general strategy that would be applicable to a broader range of models. And it's this sort of approach that we've been trying to take in my lab. And so I like to sort of give an example of the general genetic strategy that we're using by showing the Drosophila eye. So this is a scanning electron micrograph of the ommatidial eye of a fly. And in the wild type, it has this very beautiful regular structure. But you can isolate mutations in the fly that cause aggressive photoreceptor degeneration, and this ommatidial eye becomes non-functional and degenerates. But what's nice in a good genetic model system is you can do a screen in a mutant background to identify second site mutations, that is, second mutations in the genome that either enhance, meaning make the phenotype even worse, or suppress the phenotype, meaning the second mutation actually ameliorates the degeneration caused by the primary mutation. Okay. And so our strategy here that I'm going to tell you about is to essentially identify gene targets in which you can induce a suppressor mutation to compensate for or partially restore the function and prevent the degeneration caused by the first mutation. Okay. And this strategy was born of some speculation that we had a long time ago. And we had the idea that, well, if you have this scenario where mutations in rod-specific genes cause rod death, which leads to night blindness, the real problem that you'd like to solve is to prevent the inevitable secondary cone death that the rod death induces. And that leads, obviously, to a much more severe form of blindness. And we had this idea that perhaps we could exploit our knowledge of photoreceptor development to come up with a strategy to prevent the degeneration of rods. And we had the idea that we could use a knockout of NRL, a key rod photoreceptor sulfate determinant, to confer a protective effect on the rod. So the idea here is that photoreceptor precursors make a choice. They have to either decide to become a rod or a cone. If they turn on NRL, they differentiate as a rod, and the cone fate is prevented. However, if NRL is not turned on, then the precursor will differentiate as a cone. And so we thought, well, if what's causing the rod degeneration is a mutation in a rod-specific gene, perhaps if you were to acutely knock out NRL in the adult rod, that you might be able to induce it to transform into a cone and thereby make it immune to the effects of mutations in ROD-specific genes. This was a bit outlandish idea, and we weren't sure that we could achieve this transformation by acute NRL knockout, but we tried it anyway. And we took initially an approach where we created a floxed allele of NRL and used tamoxifen induction of recombination of that floxed allele to knock out NRL throughout the retina. We later then evolved into using short hairpin RNAs against NRL delivered by AAV. And more recently, we've used approaches, as have other groups, to deliver guide RNA against NRL to knock it out in animals that are ubiquitously expressing Cas9. So I'm just going to give an example because all of these approaches give a similar result in our hands of an AAV where we deliver an NRL, a short hairpin RNA targeting NRL. And so we take this AAV, we package it, we inject it into the subretinal space. It leads to a temporary retinal detachment from the RPE. And if things work well, that heals itself, it gets reapplied, and then eventually you get infection, selective infection of the photoreceptors with the virus. So this is an example of a fairly good infection where more than half of the retina is nicely infected with the virus. We include a green fluorescent protein in the virus as well, so you can tell which parts of the retina have been infected and treated. And in this case, I just want to highlight the regions that I'm going to show you in the next slides to see what the effects on gene expression are. And so we can focus on those regions that are infected, uninfected regions, and this sort of crucial transition zone. So you can see how knocking out or knocking down NRL directly affects gene expression. And so then what we do is like basically a series of in situ hybridizations. And what you can see is that in the areas of infection, that the transcript levels of NRL are reduced, and that's what we're aiming for. In areas of where it's not infected, then NRL transcript levels are normal. And crucially, if you look at ROD-specific genes like rhodopsin or GNET1, in the areas of infection, you get a marked reduction in the signal on in situ hybridization, and you can see that this is just not some regional artifact incompleteness of the in situ hybridization, but you get a very sharp transition in the transcript levels, right, where the infection peters out. So this was exactly what we were hoping to see by knocking down NRL, that it might see a large-scale reduction in ROD genes and a corresponding upregulation of cone genes in these transformed cells. And so we found initially that if you do in situs for cone genes, at least a subset of the cone genes show what we hypothesized might occur. They're upregulated in the areas where you've acutely knocked out NRL, but of course not in those areas where it's not knocked out. Now, subsequent studies, including RNA-seq profiling, have shown that indeed we see a wide ranging reduction in ROD-specific genes, but in most cases, they're not completely shut down. So when you knock out NRL during development, gene like rhodopsin is basically completely gone. Here, it's reduced more on the level of, say, 80 to 90%. Conversely, we later found that some, but most cone genes are not upregulated. So it seems that the phenotype you induce upon acute NRL knockout, this cellular reprogramming, is sort of an incomplete reprogramming from a transcriptomic level. So some ROD genes down quite significantly, a subset of cone genes up, but not a full transformation into a cone. And so we went further and used electron microscopy to try to see what was happening at the morphologic level. And I'm going to just summarize that briefly by focusing on a specific nucleus of a single cell. And many of you may know that the mouse, being a nocturnal mammal, has a rather unusual nuclear architecture in the rod, where almost all of the heterochromatin is grouped in the center of the nucleus, and the euchromatin is localized to a thin shell at the outer edge of the nucleus. In contrast, a typical mouse cone has a sort of more broken up pattern of heterochromatin, often associated with or near the nuclear envelope, and sort of a seemingly more abundant and more radiolucent population of euchromatin. And we found and quantified in this study that in the acute NRL knockout rod, the nuclear morphology looks more or less still rod-like, but the heterochromatin is broken up a little bit more, and there's some other changes. So it's somewhat intermediate between the two phenotypes. Another aspect that we wanted to look at was physiology, and again, this was done in collaboration with Vladimir and Sasha in his lab. And that is, once a photon of light is absorbed and the chromophore undergoes 11 cis retinal to all trans isomerization, this spent chromophore has to be recycled and eventually rekinked into this 11 cis configuration. And it can happen in a couple different ways. So one way is the recycling occurs via the retinal pigment epithelium, where the spent chromophore goes to the RPE from both photoreceptors and then is returned to both rods and cones as 11 cis retinal. But another pathway is via the Mueller glia, and under normal circumstances, this Mueller glial retinal visual cycle occurs by delivery of 11 cis retinol to the cone only, and not to the rod. And in collaboration with Vladimir, we found that in the acutely knocked out NRL rod, the rod acquires the ability to now utilize the 11 cis retinol derived from the Mueller glia in this side of the cycle. So in other words, then, the acute NRL knockout does reprogram the wild type rods, but not completely into cones, but into something intermediate. You might call them cods. And that this reprogramming occurs at a molecular level, where we see co-expression of both rod and cone genes, but a reduction in the rod genes. At the cellular level, where there's some sort of ultrastructural changes that put the cell in a state somewhat intermediate between rods and cones. And then also at a physiologic level, where the cells show features that are both rod and cone-like. So this result was actually not entirely what we hoped for. We thought maybe we could get these cells to be reprogrammed or transformed entirely into cones, but it ended up being something intermediate. But we still wanted to go ahead and see if this would, in fact, confer a neuroprotective effect in a model of photoreceptor degeneration. And so we initially did this with this system with a flox allele of NRL, so we could use tamoxifen induction to induce a recombinase and knock it out throughout the retina. And we did it in the rhodopsin mutant model. And the way we would do this is basically induce knockout of NRL at about 1 month of age, and then later evaluate the mice at about 3 months of age, when rod death has progressed and cone death or dysfunction has been. And what we found when we did this is that we got pretty significant preservation of the outer nuclear layer in terms of the number of cells is quantified here, as well as at least some better preservation of the inner segments of the rods. But even more importantly, I think, if you put cone-opsin antibodies on these retinas, you can see better preservation of the cone outer segments, which are virtually undetectable in the untreated rhodopsin mutant at this stage of development. And the presence of these cones correlates with residual photopic B wave, where we see very little response in the untreated retinas. So that sort of summarizes up to this point the initial studies we did. We were only working in this one model in the first paper that we published, but a number of other groups subsequently took this up, including Jijiang Wu's lab and his colleagues at the National Eye Institute. They were the first to take the approach of trying to acutely knock out NRL using CRISPR-Cas9 delivered by AAV. That proved to be effective. And they tested it in several additional models of photoreceptor degeneration and found that acute NRL knockout was able to confer a protective effect in multiple different models of degeneration. So that's where things stood. And for various reasons, my lab sort of took a hiatus on this project for a while, where some of these other groups had taken it up. And about 5 years ago, we started returning to it, and we got interested in the question of exactly like what's going on here. Like when you acutely knock out an NRL, presumably there's a lot of downstream gene expression changes. Maybe only a subset of them are actually what's conferring a neuroprotective effect in these various models. Is there any way that we can sort of parse this out and try to decipher the mechanisms and perhaps maybe then identify a sort of more targeted approach to therapy that can be used rather than using this sort of wide scale changes that occur when you knock out NRL. I should actually, though, add one other important point, and that is if you profile the effects of developmental knockout or germline knockout of NRL, you see thousands of genes whose expression changes in the knockout condition relative to wild type. However, in acute NRL knockout, the scale of gene expression changes is much smaller. So it's more on the order of maybe 150 different genes that are dysregulated. So it's still a pretty significant set of gene expression changes, but it's an order of magnitude or more fewer gene expression changes that occur during developmental NRL knockout. And so in rods, NRL plays this sort of dual role of both repressing rod gene, both activating rod genes and repressing cone genes. And in part, the repression of cone genes is mediated by the induction of expression of another downstream transcription factor in NR2E3, which in turn represses cone genes. So if you knock out NR2E3 during development, you see an upregulation of cone genes in rods and relatively little effect on rod gene expression in mice. Okay, so that leads to sort of a hybrid phenotype where the rod is still expressing all its normal rod genes, but also now is expressing a subset of cone genes. So we thought maybe we could target NR2E3 and see if by targeting this branch downstream of NRL, whether we get the same or a subset of the therapeutic effects caused by targeting NR. And so we took a very similar strategy, but this time we were mainly focusing on using the CRISPR-Cas9 based approach. And we created an AAV with an NR2E3 guide RNA and a red fluorescent protein so we could again track which cells are infected by the virus. We inject this subretinally into mice that ubiquitously express Cas9 so we don't have to bother with bringing the Cas9 along on the virus and allow the retina to reattach and hopefully get good infection of the photoreceptors. And this is, again, an example of one of these AAV injections in a wild type animal. And I'll note that we rarely get really nice uniform infection throughout the retina. Usually, more typically, we see something like this, where it's, say between 35 and 45% of the retinal surface area is infected with the virus. And this is an important consideration when you use assays to read out the extent of rescue that involve assaying the whole retina, because obviously your therapeutic effect is going to be more limited than it would be if you were achieving the rescue throughout the entirety of the retina. And so we use a similar sort of strategy, in this case in a RD10 model, where we try to do the injection of the AAV a bit before the phase of rapid rod degeneration, and then we evaluate the retinas at a time point where the rods are largely gone, and then the cone dysfunction is progressing. And so if we do this with a sort of mock non-targeting control guide RNA, this is what the data nuclear looks like. So you can see that these are cells that have been infected, but there's been no rescue whatsoever. when we do this with a guide RNA against NR2E3, in contrast, we get a pretty good cellular rescue of the outer nuclear layer. We did this with a second guide RNA against NR2E3 to make sure that this wasn't due to some off-target effect of the first guide. We see a similar rescue. And as had been shown previously, if you do this with a guide RNA against NRL, you get a comparable cellular rescue that is quantitatively indistinguishable from acutely knocking out NR2E3. And so sometimes, like I said, we can get actually a really quite decent infection throughout maybe 80 to 90% of the retinal surface area. And when we would get ones like this, we would get really quite a robust rescue of outer nuclear layer. And this is an example of an animal where one eye was treated, the other eye was not. And this is a corresponding ERG response. So there's definitely electrophysiologic rescue, and we'll go into that right now. We wanted to see if this rescued the animal's visual guided behavior as well. So we used a Morris water maze assay. And the way this works is you can take an animal and put it in a tank of opaque, basically white water that has a pigment in it. And there is a submerged platform that the animal has to find. And usually the platform is indicated in this case by a sort of red sign. And initially the animal really doesn't want to be in the water. Will scurry around until they, by chance, find this platform and they can then get out of the water. And you do this over repeated trials, and an animal that can see soon learns to associate this with this platform of safety with the presence of this object, in this case, this red flag. But if the animal can't see, obviously, then they'll continue to wander around. And so they're what we call the path length, and the amount of time it takes for them to find the platform will not improve much over time. And when you do this in animals that have been treated with a non-targeted control versus NR2E3 guide RNA, we see a significant reduction in both the path length and the latency, that is the amount of time for them to find the platform, over successive blocks of the trial. So the idea there then is that the animal is in fact having its vision rescued and able to more rapidly get to the platform. And you might argue that this change is not very dramatic, but again, after the fact, we examined these retinas, and in most cases, we are getting maybe between 35 and 40% of the retinal surface area rescued. So we think we probably get an even better functional rescue if we're able to get a greater percentage of the retina treated. So we were encouraged by that result with acute NR2E3 knockout and decided to go further and start testing this in other models of degeneration. So we went back to a model we first used with NRL, the Rho -- model, injected at P21 and then evaluated P90. And again, this is retina that was not treated. And we were a little surprised and disappointed to see that when you treat with the NR2E3 guide RNA, there's absolutely no rescue whatsoever. And that's in contrast to what was previously shown for acute NRL knockout. So NRL still rescues in this, but NR2E3 knockout does not. And then we also did this in the Rho P23H homozygote, which is a rather severe and rapid degeneration, injecting at P7 and then evaluating at P30. This is untreated. The NR2E3 knockout also does not rescue in this model, but we do see some rescue when we acutely knock out NRL. So this is a summary of those results that indicate a certain selectivity in the rescue upon acute NR2E3 knockout. Namely, we get very robust rescue of the RD10, but both of the different Rho mutant models, it fails to rescue, whereas acute NRL knockout does show therapeutic effect. So we wanted to delve into this a little bit more and see if we might be able to figure out what's going on, what accounts for the selectivity of this rescue when you acutely knock out NR2A3. And so what we did was to infect wild type retinas with AAV, either, and I say wild type, I mean they don't have a degeneration mutation, but they actually express Cas9, and inject with an AAV carrying NR2A3 guide RNA or a control non-targeting guide RNA. We then fax out the cells that are infected and perform RNA-seq, and then compare the effects on the transcriptome. And what we see, similar to with acute NRL knockout, is that acute NR2A3 knockout induces a really modest set of gene expression changes. And we saw about 37 genes that were significantly dysregulated. The vast majority of them, shown in green, were upregulated, consistent with NR2A3's role as a repressor in rod photoreceptors. And the majority of, or at least about half of these genes, as indicated by these stars, were cone-enriched genes in the adult mouse. So that makes sense. We know from prior studies that NR2A3 has a major role in keeping cone genes off in rods. So similar to the germline NR2A3 mutant, when you acutely knock out NR2A3, you see upregulation of cone genes. And we noted when we were investigating this list of genes, one gene in particular that might offer an explanation for this selective rescue of the RD10 mutant, and that is there was upregulation of PD6C. And just to kind of highlight for those not familiar with the differences between the rod and cone phototransduction cascades, most of the components in the two cascades are homologous, but actually encoded by different genes. Okay, we all know that for opsins, you have rhodopsin and you have the various cone opsins, but it's also true of the various downstream components of the phototransduction cascade shown here. And notably, in the RD10 mutant, you have a mutation in PD6B, which encodes a subunit of phosphodiesterase, whereas in the cone, PD6C encodes this corresponding subunit. And so in this mutant, PD6B RD10 mutant, that gene product obviously is gone. And it appears that upon acute NR2A3 knockout, there's an upregulation of the cone homologue of this gene. And so we speculated that this might be a good mechanism for, or a good explanation for the selective rescue of this particular mutant, because essentially you're getting a gene augmentation or gene replacement type mechanism that's rescuing. And so to look at whether the protein was actually upregulated, we used an antibody against PD6C in retinas that were infected with controlled non-targeting guide RNA. You can see the expression in cone outer segments, but when you acutely knock out NR2A3, you see kind of a cloud of PD6C expression throughout the outer segment layer here, indicating that it's now upregulated in the rod outer segments as well, as predicted from the transcriptomic study. So basically, we decided to test the hypothesis that PD6C upregulation is actually the therapeutic mechanism upon acute NR2A3 knockout. And we, this is what I'm showing here is the result where PD6C is intact in the RD10 model. And we see again, as we showed before, a very nice rescue when you acutely knock out NR2A3. But if you introduce homozygous mutation in PD6C, much of the cellular rescue induced by acute NR2A3 knockout is abrogated by this mutation. It's interesting because we still see a little bit of rescue upon acute NR2A3 knockout, even in the absence of PD6C. But we estimate that as much as 80% of the rescue is dependent on the presence of an intact copy of PD6C. So it does suggest that there are both sort of PD6C dependent and independent mechanisms of therapeutic effect upon acute NR2E3 knockout. But we believe basically that it at least accounts for the majority of the therapeutic effect that we see. And it may also help explain why it's so selective for that, the RD10 model. Now, I will add, and we were surprised to see this, but I'm not showing the data here, is we did the same experiment with acute NRL knockout, and we found that the NRL knockout rescue of RD10 is completely independent of PD6C. So we get the same extent of rescue in the PD6C mutant and wild type backgrounds. So that we didn't expect, because we thought at least a part of the therapeutic effect of knocking out NRL would be due to the downstream reduction in NR2E3 levels and corresponding upregulation of PD6C. That doesn't appear to be the case. And so this is sort of a summary of what happens when you knock out NR2E3 acutely. You see mainly an upregulation of a couple dozen cone genes in rods and no appreciable effect on rod gene expression. Acute NRL knockout, in contrast, causes a similar upregulation of cone genes, but also downregulation of rod genes. But somehow the differences in the downstream gene expression between acute NR2E3 knockout and acute NRL knockout lead to pretty striking differences in the sort of range of the therapeutic effect that you can achieve. I'm not going to present any data on this topic, but we've been collaborating with a pharmaceutical company, Ionis Pharmaceuticals, to try to develop antisense oligonucleotides that can target NRL to see if we can achieve a similar therapeutic effect, but with a more translatable approach. We now have some pretty good antisense oligos against NRL that can reduce the levels of NRL as much as 90% after intravitreal injection. And it really lasts quite a long time. So 3 or 4 months later, you can still see some appreciable reduction in NRL transcript levels. We also can see the same downstream gene expression changes that we see when we knock out NRL acutely, either by AAV-delivered CRISPR-Cas9 or by tamoxifen induction of recombination of a flox allele of NRL. And lastly, and importantly, it appears that these antisense oligos against NRL also can give the same range of therapeutic effect that we see with acute knockout of NRL. And what I like about the approach is that we can really target all the cells, unlike with the AAV approach, where we always get only a subset of the cells rescued here by intravitreal injection of ASOs. We can really get a rescue across the entirety of the retina. And we're trying to now develop similar ASOs to target human NRL. So these are the people that participated in the work. It was initiated by Cindy Montana almost 10 years ago. Actually, she's now an ophthalmology faculty at Washington University. She did this work when she was an MD PhD student in my lab. And the current work, especially the NR2E3 work, is carried on by Dan Murphy in my lab, who's a staff scientist and lab manager. And of course, this work has been done in collaboration with Vladimir and Sasha in his lab.
Joe, I have a quick question. If I understood what you said about the company, you're doing this intravitreal injection?
Yes. Yeah.
So how that it just penetrate entire retina?
So I was contacted by Ionis maybe 2 years ago, and they said they had been developing this. And to be honest, I wasn't that familiar with antisense oligos at that time. For those who aren't familiar with them, antisense oligonucleotide technology is usually a kind of an unusual DNA-RNA hybrid molecule with modified RNA nucleotides that sort of extend the life of the molecule. So it makes it resistant to normal degradation. They still get degraded eventually, but they perdure. And for reasons or by mechanisms that I am entirely ignorant of, if you inject into the vitreous, they go right through the retina and get into the rod nucleus, and they stay there. And I was very skeptical until we saw the effects on downstream gene expression, and we knew it really had to be getting into the rod nucleus.
And second question I have, NRL germline deletion produces this rod-like, cone-like photoreceptors, but they are unstable. So they degenerate maybe slower than, let's say, RD1 or RD10, RD10. So do you observe whether there is a change, how stable they are after your rescue?
Yes, so it's true. Germline NRL knockout in mouse leads to a sort of a relatively rapid phase of degeneration in a small subset of the cells, and then a very, very slow degeneration in the remainder. So if you go out, say, a couple of years, you'll see appreciable degeneration. And our therapeutic effect does perdure, but over time, if you take much later time points, then we do see progressive thinning of the ONL. So yes, it's not a perfect preservation. They do slowly degenerate.
Really nice talk. So I have just curious for the COG, right? So where I also is important for master regulator. You repress it, the cone genes go up, and then you have this COG. So if I'm just wondering if once you reach that COG state, if you remove the artificial repression of RNL, what happened to the cell? Do they revert back to rods or they stay at COG?
So you're saying if you were to repress
like the oligo,
antisense oligo.
Yeah, exactly. Yeah.
That's a really cool question. Like whether there's a sort of permanent epigenetic state or change, but we haven't gone far enough to test that yet. So I'd really like to know that, because if this were to ever be used for therapeutic purposes, the reason I like the antisense oligos is it is potentially reversible, right? So if you see some adverse effect, you can discontinue therapy, and hopefully then the cell would at least return to its prior state. Whereas, of course, if you use a knockout approach, then it's going to be a permanent change. So that's a really interesting question.
Hi, Dr. Corbo. Very nice talk. I have a question related to the first part. As you go deeper underwater, you lose the long wavelengths. So why do you want to shift red to even redder? Do you see a red shift in a UV photoreceptor?
Okay, so you're referring to the first part of the talk. So it's a really interesting phenomenon that when you switch the chromophore from A1 to A2, the effect is much greater on the longer wavelength opsins. Okay, so you get a 30 to 40 nanometer shift of the red cone opsin when you switch A1 to A2. But the shorter the normal lambda max, the A1 lambda max of the opsin, the shorter the red shift, such that when you get down to the blue and UV, it's only a few nanometers. Yeah, so that phenomenon, there's some sort of complicated biophysics that may explain that phenomenon that I can talk to you about offline, but it basically has to do, it's thought to have to do with the extent to which the ring of the chromophore is coplanar with the conjugated system in the non-ring part in the linear system. And the less coplanar it is, the less you get the delocalization into the ring, and therefore the effect of changing and adding another double bond in the ring is correspondingly less. And so the longer wavelength opsins, the ring and the chain are essentially coplanar, and they become less so in the shorter wavelength opsins. That's one theory to explain it.
Hello. I really appreciated your talk and your innovative approach to. I appreciated your talk and your innovative approach to addressing diseases like retinitis pigmentosa. My question is about one of your previous slides in which you showed a table of other genes that seem to have kind of a downstream effect when you messed with the I'm blanking on the name of this.
NR2E3.
Yes, that gene. In that table, there was a gene called adrenergic receptor beta 1. And my question relates to the oligo antisense nucleotides and how if you inject that intravitreally and possibly affect that adrenergic receptor beta 1, you could actually start messing with the pressure of the eye. If you overexpress it, you would likely cause increased pressure. And have you noticed any of the eyes treated with that would have like?
But these gene expression changes are presumably only occurring in the rod.
Oh, okay.
Right. So I'm not sure it would be expected to have that effect, but it's an interesting thought. We haven't looked at pressure at all.
All right, we will move to Zoom.
And we have one question from Steve Lisler.
Oh, hi, Joe. Great talk. My question was, when you target gene manipulation in a rod or in a cone, does it affect the next order neuron or the third order neuron gene expression?
The shorter answer is we haven't looked closely at that. But that is a very good question. We've often been asked whether the connectivity of the transformed rod with second order neurons, bipolars, or horizontal cells is changed, which is also something we haven't looked closely at. Oh, great question, but we ought to look at it.
And Daniel Palanker. Daniel, are you still there? Okay. Gordon Fain, would you like to make your comment?
Oh, I didn't need to make it audibly. I just wanted to point out that the reason why the shift occurs in the pigment is because in murky water, the animals see better because light scattering is inversely proportional to I think the fourth power of wavelengths is very heavily dependent upon wavelengths. So red light scatters much less in murky water, and animals with redshifted pigment see better. I thought that was sort of the previous question about the point of the transformation. That's all. No question. Gorgeous seminar, Joe, as always.
Hello, Dr. Corbo. Great talk. I'm Rafał Pazuk in Polski Lab. I have a question. Do you have any data how that acute gene knockdown would translate to cone-dominant species like squirrels, monkeys, humans?
When you say translate, you mean like if we were to acutely knock out NRL in the rods of those species?
I'm asking if that would slow down the degeneration too in cone-dominant species.
I'm not sure, honestly. I mean, I guess it hinges in part on what's the mechanism of secondary cone degeneration, right? And in animals like a mouse or a human, where the rods are enormously dominant and constitute most of the cells in the outer nuclear layer, I suspect when you have massive loss of those cells, then you're really changing the cellular environment in the outer nuclear layer. You're obviously getting a lot of reactive changes, gliosis, and introgression of inflammatory cells, and probably other changes. I know some people have speculated on the loss of factors released by rods that are protective or nutritive for cones. That's another possibility. But I tend to favor the idea that the outer nuclear layer becomes a very inhospitable environment when you lose all those cells. Now, in an animal where the majority of the photoreceptors in the outer nuclear layer are cones, usually in many of these animals, like the squirrel that you mentioned, the outer nuclear layer is actually very thin. It's only 2 to 3 nucleotides, 2 to 3 nuclei thick. And so loss of rods isn't going to cause the same cellular disruption at the outer nuclear layer in those cases. But I just don't think people have systematically studied this in such species. So it's hard to know.
Can I offer an alternative explanation for the salmon shifting wavelength?
Of course, please.
I think as someone who's a fisherman, salmon in the oceans swim in deeper water, surrounded by blue, whereas if we migrate, they swim in shallow water, surrounded by mud and trees and woods. So it's, I think, simply a matter of, let's say, depth of water. So if you half a meter deep in the water, you have more web life. Red light.
Absolutely. This is totally true. So it's well known, even in completely pure water, shorter and longer wavelengths are preferentially absorbed. And so if you go down and say to 100 or 200-meter depth, the predominant wavelengths are centered around 480 nanometers, 490 nanometers. And most of the shorter and longer wavelengths are filtered out by there. So that's exactly true that the salmon in the open ocean are at these greater depths. They go into inland streams and are often in extremely shallow water, less than a foot or two, right? And so those longer wavelengths certainly penetrate there better. So the whole point is that the light environment is shifted, and there's a lot more available light at the longer wavelengths that they can take advantage of.
Hi, Joe. I just have a simple question. Have you tried to acutely knock out NR2E3 in heterozygous P23-ish model? I'm asking because in more recent data in a germline knockout of NR2E3 in that model, we see a difference compared to homozygous.
In the P23H hat, you said?
Yeah.
We haven't done that yet. But
okay, maybe we can.
Yeah, we should. We should talk about it.
Yeah. Hi, I'm Kaipa from Jodas Lab. So what is the delivery vehicle for the ASOs?
I'm sorry, I didn't understand.
The delivery vehicle for the ASOs.
The delivery vehicle, how is the ASOs are delivered?
They're injected straight out. So they're not in any, lipid or anything like that. They're just the naked ASOs. Yeah.
Yes, thank you.
But I think they have proprietary technology for ASOs.
It is. Yeah.
Right? Is it like a fatty acid conjugation, right?
It may be. Yeah. They can't tell me.
Used to be called
I don't know the exact chemical details of the way they make them.
Used to be ISIS before, right? So
Correct.
Hi. I'm just wondering if, in addition to NRL knockdown, you've thought about a combinatorial approach with like overexpressing pro-cone differentiation transcription factors such as CRX.
Yeah. So the idea is, can you push the cellular reprogramming all the way to a cone? So we've thought a lot about this, and we have a list of cone factors that would be good candidates. I mean, we haven't actually implemented the approach, but we've thought about taking a screening approach, sort of similar to like the way Yamanaka did to first characterize reprogramming factors. So combine acute NRL knockout with misexpression of one or more pro-cone transcription factors. I think that would be the right strategy. Perhaps also combine it with some factors that might sort of loosen, the epigenetic structure in some way. But we haven't tried it yet.
And I was also curious about, I was curious about if there are regulators similar to NRL in zebrafish that would change the population of cones and rods, and how that could potentially affect the patterning.
When you say regulators in zebrafish similar to NRL, I mean, the rods express NRL in zebrafish. They have a homolog of NRL. So NRL or NRL is a member of a family of transcription factors called MAF transcription factors. And for example, birds don't have NRL, but they have another MAF factor in their rods called MAF-A. So almost all vertebrates use one MAF factor or another. In fact, we recently found that even lamprey rods express a MAF transcription factor. It probably plays a similar role. So it's likely that MAF factors are involved in rod differentiation in all extant vertebrates, and it probably dates to the common ancestor of extant vertebrates. I don't know if that answered your question exactly.
I was sort of curious about how that impacts the patterning, like the mosaic.
So, many fish species like zebrafish are cone dominant and have these very regular mosaics of cone photoreceptors, and the rods are sort of interspersed in between. I don't know. I don't think, the effect of knocking out factors in the rods has been studied quite systematically on the effect of the cone mosaic. We have studied zebrafish knockouts for a number of transcription factors that affect cone development. Those can really perturb the mosaic, but we haven't really tried targeting rods.
Super talk. I just have one question. So when you convert the rod into cones-like behavior, but you still have the mutation like PD6, right? So does it actually, when you convert them, does it not require them, or is it, how do you, because you still don't deal with the loss of function, right? So how do you put that in mechanistically? How do you see the rest?
Well, so the rods, you have to keep in mind that I think that the functional rescue is more the preservation of the cones, right? And the rods themselves, like when you acutely knock out an NRL, are not very active because you've reduced the expression of rhodopsin, but you're not upregulating any cone opsin. So there's not a lot of opsin in the cell. So that's an arguably a downside to this approach, is you're sort of inactivating the rod, maybe putting it into some sort of state of dormancy for the sake of preserving the cones instead.
Gordon, would you like to have a follow-up comment?
Yeah. Joe didn't talk about lamprey. But lamprey are interesting because lamprey are anadromous just like salmon. They also use this exact same mechanism, the same protein to convert between A1 and A2. When they come to breed, the reason I find that so interesting is because salmon, diverged from all other vertebrates 500 million years ago. So there were apparently this mechanism, it's the same protein also expressed in the RPE. It's difficult to believe that it wasn't, that this isn't a very old mechanism dating back to the origin of the vertebrates. And so what that means is that vertebrates were penetrating lakes and streams way earlier than any vertebrate evolutionary biologist would tell you that they were. And I find that fascinating. But that's really all I.
Yeah. We've been doing a number of comparative studies with lampreys, aside from the story with CYP27C1 and the A1A2 switch, and there are quite a lot of striking homologies between the photoreceptors and photoreceptor types of lampreys and those in jawed vertebrates. It's quite interesting. So we're slowly developing a picture of what the common ancestor, the complement of photoreceptors and the common vertebrate ancestor may have looked like. And it appears that it may have had a similar A1A2 switch.
Yoshikazu Imanishi, are you ready to ask your question?
Can you hear me?
Yes.
So my question is that what is the advantage of the NR2E3 knockout compared to this NRL knockout in the acute setting in terms of therapy? So does NR2E3 knockout confer meaningful vision, cone-like vision to those mice?
Well, it does appear to confer protection on the cones because we get some functional rescue. But I think its therapeutic utility might be more limited, especially if in human acutely knocking it out has a more limited therapeutic effect. I will say, however, that the effects of mutations in NR2E3 in human and in mouse are quite different. So it's likely that the downstream pathways have been rewired to some extent between the two species. And so I would say we kind of would have to do more investigation in human photoreceptors to see the full extent of the gene expression changes. And so acute NR2E3 knockout might very well have more therapeutic potential in a human than appears to be evident in mouse models.
Hey, Joe, thank you for your talk. A couple of questions. I noticed in your acute NRL knockout that the retina seemed to develop rosettes. So can you comment on that?
Yeah, so we've been asked about this before. And people who have studied the germline knockout of NRL or NR2E3 have observed that you get striking rosette formation in the outer nuclear layer.
Mm-hmm.
Overall, Vladimir, however, a lot of the so-called rosettes that we see, especially after a subretinal injection, often involve sort of the whole thickness of the retina. They look more like sort of retinal buckling than rosettes of the outer nuclear layer. In other words, I think they're quite distinct morphologically from what you see in the germline knockout. And in our original study, where we used flox allele of NRL, where it doesn't involve any intraocular injection, we don't really see that.
Okay.
So there's a little waviness of the outer nuclear layer, but we don't see the frank rosettes that you get in germline knockout. So I think this is a distinct phenomenon, probably caused by the subretinal injection and incomplete or imperfect reapplication of the retina to the RPE.
Okay, thank you. And my second question is very generally, can you comment on the current thinking of the difference between hetero and neurochromatin and how that, why rods and cones are different and how that might affect gene expression?
There have been several studies about the purpose and the mechanism of the so-called inverted architecture of the rod nucleus in nocturnal mammals. And the general thinking is that it has a role in changing the refractive index of the outer nuclear layer. So nocturnal mammals tend to have a rather thick outer nuclear layer through which, of course, the light must pass to reach the outer segments. And that by having this inverted architecture, you essentially are changing the light guiding properties of the outer nuclear layer and permits more direct passage and less scattering of light. That's the belief.
What about gene expression? Could that also regulate the efficiency?
Yeah, so what's the effect of nuclear inversion on gene expression? That's less well known because you would, somehow want to figure out a way to induce a mutation or a change that caused loss of the nuclear inversion, but without changing anything else. And it's hard to know how you might do that.
Thank you.
Joe, did you sequence or do you know the efficiency of a Cas9 inactivation? The efficiency.
Of the how quickly?
No, not how quickly. What percent?
Oh yes, I'm sorry, I didn't
in...
show that. So when we deliver the guide RNAs for either F3 or NRL, we get about 90% knock. 90% of alleles are knocked out. Yeah.
Beautiful talk. And in the first part of the presentation, you showed us the enzyme that converts the vitamin A into this other form is localized in the mitochondria. And when you think about the visual cycle, the flux of vitamin A stays confined in membranes and in the endoplasmatic reticulum. So my question to you is that, do we know of any protein that is actually channeling this vitamin A into the mitochondria preferentially?
I don't know the answer to that.
All right, we have last three questions, the toughest. Are you ready?
I think I'm ready.
All right.
Not at all. Hi, Dr. Corbo. Thank you so much. I thought it was wonderful. So I wanted to ask, is there any benefit to having this rod characteristic genes knocked out or deficient, such as the row background for NRL knockout? So maybe that would aid the transition into this rod cone intermediates, perhaps? Instead of having a different background where you have like the PD6B, the RD12.
I'm not sure I understand. You're saying if you acutely knock out NRL in a rhodopsin mutant, does it make this?
So question was perhaps if you have this multiplex editing, so you're knocking out both NRL and a rod characteristic gene, so maybe a rhodopsin, that would aid the transition into these rod cone intermediates.
Well, we haven't tried that. I'm not sure whether that would aid a transition. I suspect it wouldn't.
I really loved the morphology sections that you showed in the first few slides. And there is a question that I have for a long time, and I'm hoping that as an expert on connection, maybe you will help me. So the cones nuclei in the outer portion of the outer nuclear layer, what is contributing to that?
Why are the cone nuclei usually at the outer edge of the ONL? That's a very good question. That's the case across most vertebrate species. There are only fairly rare exceptions. So whenever you have it's hard to tell if you have a species where the ONL is only a couple nuclei thick, then usually it's mostly cones and they're all there at the edge, but the whole thing is the edge. But when you have a thick outer nuclear layer with lots of nuclei, let's say 10 to 12 nuclei thick, then the cones usually predominate at the outer edge. So there's actually a number of studies that have been done. Mutations in a variety of genes will cause aberrations in the localization of the cone nuclei. And also in the mouse, the cone nuclei are actually more evenly spread throughout the outer nuclear layer in the first postnatal, the first 10 days postnatal, and then they all rectify to the outer edge. So it's a controlled process for sure. And some genes are now identified that are required for the process, but why they do it is a different question, and I don't know the answer.
On your in situ, when you show, there is like a very dark layer close to cone nuclei, and this is rod probe instead of being throughout the entire layer.
That's
So
that dark
why is
layer
that?
for like a rhodopsin in situ. That dark layer is in the inner segment. So most of the for many transcripts, they're most concentrated in the inner segment of the rods.
Thank you so much for that really interesting talk. So I was kind of curious, do you have any data on like doing variations in viral titer with the NRL knockdowns? I was kind of curious if there are effects of transduction mosaicism in terms of like, is it more advantageous to have a specific ratio of like NRL positive rods left, surrounded by kind of transduced NRL knockdown rods?
That's a really interesting question. That's hard to explore because it's hard to really control. I guess, like you said, we could change the titers of the virus. But another way to kind of ask that question is to maybe change the amount of antisense oligonucleotide that you deliver. You could sort of lead to different extents of reduction within all the rods. And I think that might actually be advantageous. I mean, the data is still early, but I think in some cases with the ASOs, we're getting actually better rescue than when we do the complete knockout with CRISPR-Cas9. And it could be that having a little residual NRL is actually beneficial to the cell.
Thank you.
Yeah.
All right, please join me. Thank our speaker today.